How about now? How about now? Is it working?
Yes, it's working.
Oh, great.
The audio is not working again.
All right. I think I have to use the internal microphone on the camera. On the computer. Is this better?
Yeah, this is good.
Yeah. All right. I don't know why. Maybe this microphone is somehow not turned on or something. All right. So sorry about the delay. So we're going to continue talking about phylogenetics, talking about how we interpret phylogenetic trees, and specifically, we're going to be talking about kind of how we use phylogenetic trees in order to talk about changes in genetic code and kind of what are the equivalent of how are we changing DNA and how are we representing that in a phylogenetic tree. So we were talking last class about homology. So this idea that there are a lot of shared traits which are being passed down and consistent through lineages of organisms. And so if you think about it, all of these bone specializations, all of these things are all being done by genetic code, right? And switching of slight genes. So how long, say, the radius of a species is compared to the ulna is all based on genetics, right? And so changes in what genes code for each of these different bones can lead to drastic changes in organismal body plan and things of that nature. And so, go away. All right, I guess I'll just have to close that. So as you've learned before, there are a lot of different genetic changes that can occur or errors that can occur in a gene sequence as DNA is being replicated, as DNA is being yeah, DNA is being replicated, and as it's going from, you know, one stage to the next, right? We have different types of things, right? We can have deletions of specific amino acid sequences. We can have insertions of different sequences, as well as different types of, you know, individual nucleotides. And so generally what we're doing, if you remember in class, we did the extraction of your DNA from your bacteria, and then we ran a blast search, right? We looked up the type of DNA sequences we had for a particular gene. In our case, we were looking at that 16S, and we were comparing how many differences there were in these gene sequences of your species and then other species that are known, right? And so what happens is, as species are evolving, as you know, slowly and surely changes in DNA are happening, there are insertions and deletions that can occur, right? And so what you have is, say, down here at section 3, you have this code that is at the top, this CC, AT, CA, etc. And so what happens is that over generations, we're going to have insertions and deletions. And so what was once, say, a gene for spider silk, or say the length of your arm, has now had additional nucleotides added into it, which have made it longer. And so, you know, if you have something like, say, this insertion of a GTA, you know, you're actually adding an entire amino acid that's going to be coded for there, right? And so you could drastically change the structure of that gene by just adding a new type of amino acid. And it's going to change the way the chemicals react. It's going to change its properties. And so when you do this blast search and things like that from your experiments, what it's doing is it's trying to line up these consistent parts of the genome, right? It's trying to figure out that over time, we've added a few of these. And so this is what it means when it says that something's like 99% aligned or 98% aligned. It's how much it has to move any particular part of this DNA sequence. And so that's also another reason why blast searches take a while, why you need a decent amount of computing power, is because in that database, what it's actually trying to do is it's trying to do all these combinations, right? It doesn't necessarily know that, for instance, that the gap is between CCAT and CA, right? So it's also going to try different combinations of putting gaps in between CCAT and all of those good things, right? But nowadays we're been comparing DNA, and so we're trying to align sequences from species that have been studied and then new species, and so we can document those brand new species, but we can also see how genetically they vary, right? And so the closer species are related, the less amount of these genetic differences there should be in their genome, right? A lot of really, really dramatic insertions and deletion will change things, may kill the organism. But when you are looking at, say, a lineage slowly over time, there should only be one or a few of these changes over time. And so scientists can actually estimate how far apart species are from one another by looking at how many of these mutations and errors there are. And so they accumulate over time. And so more distantly related genes will have different bases or even different lengths. And so insertions and deletions constantly. And so scientists kind of look at genes and they try to estimate how long they've evolved based on this. How many changes does it take to get from point A to point B? How many genetic changes are there in the silk that's produced by a tarantula, you know, a very early species of spider, versus the ones that you'll find on your porch, which have much more elaborate silk, right? And so scientists have this idea of what's known as a molecular clock, or a molecular timeline that they estimate based on these genetic sequences. Obviously, like everything in biology, this can be much more complicated. And so certain genes, you know, are more likely to change than others. And so One thing I want to talk about is when we are looking at these phylogenetic trees, right? How do we put them together, right? There are a lot of different variations depending on what physical features you look at, depending on what specific gene sequences you look at. These can all be arranged in very, very different ways. And so phylogenetic trees are based on what is called maximum likelihood, or it's a procedure that's deciding what is most probable. And the idea behind maximum likelihood is maximum parsimony. And so this was originally done when we looked at character-based interpretations, looking at morphological interpretations. And so the idea is that this approach attempts to maximize the number of evolutionary steps that require to explain, the arising of a particular taxon or features. And so what that means is that, the simplest explanation is the easiest. It's a lot easier to lose a trait than it is to necessarily evolve a trait, right? One is removal of a gene, one involves the complex creation of a gene that also doesn't mess with like the natural order of that organism. And so there are a lot of different ways you can do this, but when a scientist creates a paper on phylogenetics, what they're doing is they're actually looking at how many steps, how many different types of evolution or trait changes does it require in order to look at these species. So for instance, looking at this this evolutionary lineage here, can the people on Zoom see this Safari nonsense that's blocking my screen, or is it just on the? All right.
No, it's not.
Oh, okay. All right. So you look at these two hypotheses, right? 1 and 2. We're looking at how different organisms could have evolved in their lineages, right? Specifically, we're trying to look at when organisms evolved a bony skeleton. And so if you look at the top and the bottom, these are two hypotheses of trees, right? Which one do we think is the one that follows maximum parsimony? Which one involves the lowest amount of new traits? All right, so when we look at these two trees, if you remember, each of those red lines are going to be a new trait that is evolved in an organism. And so when we're looking at these, we're trying to describe the evolution of vertebrae, bony skeletons, extra limbs, hair, etc. When we look at the two, one of them has a relation, and it's harder for you because you can't see the labels at the top. But when you look at it, you can see in hypothesis 2, in order for this relationship between rays and sharks to make sense, in hypothesis 2, you actually have to evolve bony skeletons 2 different times, right? So when you look at the sharks and the rays, they could either be separate lineages. So sharks are off on the left here. Then we evolve bony skeletons, and then rays evolve, and then we evolve these limbs. Now, it's totally possible that vertebrae evolved, and then independently, rays and fish, these ray fin fish, so like skates and stuff, evolved bone, and then this other lineage evolved bone, and then evolved 4 limbs, right? That's totally possible, but it's much less likely and it's much more difficult, right? Convergent evolution of these two sets of organisms, both evolving bony skeletons, is much less likely than, a common ancestor of rays and all of these other organisms having evolved bony skeletons, right? That requires less transitions. And so this is, I would consider, one of the simplest versions of that kind of maximum parsimony. But if you think about it, this could be done for many, many different traits. This can be done for when we incorporate molecular data and gene sequences. We can involve transitions in, say, the bones that make, or the genes that code for your vertebrae or your skeleton type. And so in this case, hypothesis 1 would be the one that is most parsimonious. The least amount of these, little red transitional areas, and the fact that each of these traits only evolve once. And so I'm never going to ask you to make a tree. I just want you to understand this idea of maximum parsimony and the fact that we're making educated guesses. All of these, once again, are hypotheses about how organisms could have evolved. And so in each case, we're trying to look at the relatedness from a group of observations. But again, none of them are perfect. Phylogenetic trees are constantly being updated. For instance, when I was taking my exam for candidacy for my PhD, there had just been a paper that included molecular data and completely changed the phylogenetic tree that my professor who I worked with had published just a few years later. So when I talked to him, I said, which one do I explain for my defense? And he said, you have to pick one and you have to be damn sure that you believe that one and can defend it. And so, he was like, my work versus someone else's work. And so again, they're all hypotheses and they're changing over time. And as we get more data, we're going to add to it. And so, new sequences of DNA, discovering new species can really upheaval and change, how these are related. So for instance, in spider genomics, spiders have actually gained silk and then gained advanced silk and then lost the ability to use that silk. So for instance, things like jumping spiders are actually not, an ancestral, smaller tarantula. They're actually a spider that learned how to make silk, learned how to make a web, and then they evolved and lost that. And so this can be very complicated. And so I'm not going to ask you to be able to do this, but when we are looking at, say, a program and using a program to create a phylogenetic tree, we create something that's called a character table, right? And so this is usually yes or no, 0 or 1. And the idea is that you have a bunch of different traits and you tell the computer, does this set of organisms have all of these traits? And so does this organism have these set of traits, right? So the program will say, all right, so, organisms with hinged jaws, you'll say that bass, frogs, turtles, leopards have all those traits. 4 walking legs, and you'll tell them which ones it has. And so what it'll do is it'll try to combine and make sure that most parsimoniously, how are all these traits related? And so this one is the simplest scenario that you can have, because if you look at it, no traits are shared in a strange way by multiple species. So for instance, there's the bass, does not have any hair. But if you say had a bass that had hair on it, this would be a little bit more complicated, right? When did it evolve hair? Did all these other organisms lose hair or something like that? And so this is the easiest way for a computer to kind of analyze it. And so if you think about it, when it's putting together this tree, this lancet outgroup does not have any of the traits, right? So it has to be the most distally related organism. Then we have one trait that seems to be held by almost everyone else. So, right, the backbone. So you got your outgroup, then you have the evolution of this vertebral column or your backbone. Then we're trying to organize it. Well, the only organism that doesn't have a backbone here is a lamprey. So that must be the furthest evolved. And then we're going to look at a next trait. So hinged jaws. So now everything here should have hinged jaws. But again, this can be very, very difficult if you're using genetic features versus physical features. You can also add weighting. So for instance, certain scientists, it's harder to evolve a new silk sequence. So that's a little bit more important to make sure it's placed in the right position than say something such as, hair, which could have evolved in many different ways and many organisms have different versions of it. But again, I can ask you to interpret these just to understand that this is how we put them together when we're making phylogenetic trees. And so again, this is another example of how it's a hypothesis. And so when you look at this, this is a little bit more specific to how it actually occurs, right? So we have 3 different species of bird. And so we have something that we know is actually an ancestral sequence, right? Let's say we have a species of bird that we know existed 1 million years ago, and it had a particular coding for its feathers, right? And so we have this gene sequence, AGTT, that's what it should have been. And now we're looking at how did these species evolve such that they change their genetic sequence. So, right, species 3 has only one of these alleles the same. I mean, only one of these nitrogenous bases the same. Species 2 also only has one, and species 3 also, species 3 actually has 2 of them. And so when scientists are making these trees, these are all the different versions of these trees. And when you look at them, these trees here have 6 different changes in order to create these sequences from this sequence. And these have 7. So we're not sure which is correct, right? So these would be the most parsimonious, the ones that only take 6 events. Yeah, these 6 events, but we're not actually sure about that. Once again, these are all hypotheses, but this is how it gets very complicated when we're comparing even a single gene sequence, right? When we do a lot of these comparisons, we're not actually taking an entire genome and comparing it, right? That's too much information. So what we're doing we're taking specific genes that we know should be shared by organisms. In this case, they should have some form of this ancestral feather sequence. And then we're making these educated guesses to try to figure out these changes. And so a lot of these trees can be equally true. And that's when scientists will take in their extra knowledge or things that can't be directly put into this, such as location, maybe some connection to fossils, but they'll draw on more conclusions to try to pick this up. And again, a lot of things like this ancestral sequence may only be available in an organism that is dead or DNA that is left behind from a long time ago. And so I want to talk about some kind of distinctions in phylogenetic trees. So for instance, there's the idea of something called a monophyletic group. And so this is a species of spider that I study. I study this subfamily known as Pseudorachni, which are these cool moth specialist spiders. And so right here is one of them. It's called the bolus spider. What it does is it actually makes a single glue droplet, and this is its web. This is what it uses to catch prey. It releases pheromones, it causes moths to fly towards them, and then it throws this little glue droplet at them. And so most spiders are really bad at catching moths. And so all of these spiders are monophyletic, which means that they have a shared trait that is only found in their group. And so a shared derived character is unique to a particular clade or group. And if you remember clade taxon, these can be very large things. So for instance, when you talk about monophyletic group, all mammals are monophyletic for hair, right? Other organisms aren't going to have that exact thing. Or, marsupials and up are monophyletic for having, live childbirth. It's something that's held by an entire group. It can be something that's very specific and small, or it could be something that's very, very broad. Again, depending, you have to pay attention to the context of how you're talking about it. So I'm going to show you a video on the bolus spider because it's my favorite.
This may look like a bird dropping, but that's just a disguise to fool anything that might. This may look like a bird dropping, but that's just a disguise to fool anything that might want to eat it. In fact, it's a spider. and one with an even more extraordinary hunting technique. It's a bolas spider. Throughout the day, she remains motionless, but when evening comes, she prepares for action. She abandons her disguise and starts to move. Slowly, she makes her way down to the underside of the leaf. There she hangs from a horizontal thread. Next, she starts to spin a single strong line, pulling it out of her spinneret with her back legs. And at the end, there is a sticky globule. This is her bolas. It's all she needs. She climbs back up to her leaf and takes up her position on the horizontal thread with her weighted filament dangling from one of her front legs. A moth. She whirls her bolas, but misses. But she has ways of enticing the moth back. She can produce a pheromone, a chemical perfume that the moth finds irresistible. What is more, she can change it to suit the particular species of moth that happens to be around. The moth comes back. This time, she's got it.
Yeah, and so that moth will do that that spider will do that multiple times a day. They may even wrap up that moth and they'll kind of hang it like a fish, and then they'll keep looking for more. But yeah, so this glue is actually something that's very special. That's evolved in only a particular subspecies of spiders, the ones that learn to feed on moths, right? So they have this special glue that actually helps break over the scales of moths. So something that's you may have not noticed, or I always ask this question: have you ever played with a moth before? You might find that there's I get that look. There's always. So if you ever touch a moth, they have this powder that comes up on your hands, right? So they've actually evolved sacrificial scales that cover their body. And so these scales give a multitude of evolutionary advantages. It's one of those things that scientists can't really pinpoint as evolving for one particular strategy because it does so many things. It helps them with spider webs. It lets them have camouflage, pheromones. It helps them clean themselves. These scales are really, really useful. But they're very loosely attached. So what happens is when a moth hits a spider web, the glue sticks to the scales, the moth thrashes, and that powder comes off and they can escape. The spiders that I study, however, have a monophyletic group. They're a monophyletic group for this glue that actually overcomes the scales. It actually, instead of attaching to the scales and having them loosely flake off, it actually gets sucked underneath them, and it's specially made for this. So let's see. So for instance, when a regular spider tries to catch a moth, this is what you see on the left. All of these scales here attached to the silk thread, and the adhesion strength is just these little threads. How strong can you be with just being attached to these scales? When you look at those spiders I study, though, the glue droplets not only spread further, but they actually glue all the scales together. So it's actually it's a miracle of glue interacting with the topography of the moth scales. And so pulling this entire thing up, an entire brick of scales is going to be a lot stronger. And so that allows this species to specialize on an organism that usually is not being eaten very much in the environment, but is readily available. And so pseudarachne, these bolus spiders, have this special glue that allows them to feast on something that other things can't. And so, as I said before, these spiders are an example of a monophyletic group, right? They're all organisms that share a same trait. And so if you remember, make sure that you remember that, you know, each of these things, clades, taxon, monophyletic group, can all be at different levels, right? So for instance, this group here, monophyletic, say for that spider glue, but you could also have, say, this entire group, you know, this common ancestor here, they could all be monophyletic for something else. Say, you know, if these are spiders, monophyletic for spiders for silk, you know. So it can happen at many different levels. You then have this idea here of what's known as a paraphyletic group, which is a group that consists of some type of ancestral species and then some of its descendants, not necessarily all of them, right? So for instance here, we have this common ancestor of D, E, and F. So when we're looking at this group, we're going to talk about this common ancestor and then D, E, and F, just this group. So that means it's not monophyletic because you're missing, some species aren't being represented here. So in paraphyletic, you're essentially removing something that we know about that exists further down line. And then you have polyphyletic, which means that you include distally related species without necessarily talking about their most common ancestors, right? So in this case, you know, it's monophyletic, except we've removed some species that exist. Over here, we're no longer talking about this ancestral species or this ancestral species. We're also ignoring all of this. And so what we're doing is we're taking groups, say a monophyletic group, and then a distally related other group. And so, you know, these clades are nested into larger clades. So depending on what you're looking at, what you're talking about, it'll be a little confusing. But once again, paraphyletic, ancestral species, and not all of its descendants, polyphyletic, distally related groups without including their common ancestor. So for instance, for D, we would have to include this species here, right? Because the most common ancestor of this entire group and this D would be back here, right? They branched off after that. And so this would be the common group, the polyphyletic group. And so when we look at these spiders, so these are all the different spiders that I study, right? And this is a little graphic of how they catch prey. So this is the bolus spider we were talking about, right? They use that single droplet to catch moths. Then we have these excavata, which is pretty cool. They're like bolus spiders. They release pheromones, except they don't even care about glue. They don't make a bolus. They literally secrete pheromones, and then they wait for a moth to come by, and then they just try to grab it out of the air. They've completely given up on making silk, but they have the silk glands. So they have to be more closely related, say, to bolus spiders than jumping spiders, right? This is where it can get very confusing because not only did they lose their ability to make a web, but this species also lost their glue droplets completely. And so, but still, this is a monophyletic group, right? So then we have Placylobus, which is actually the spider that makes a pizza pie sliced web, and then it just has glue droplets on this silk. Then we have Pseudarachne, which makes a horizontal web that looks like one you'd see outside. And then, you know, as our outgroup here, we have normal orb weavers, right? The things that you would find on your porch, things that you would find outside, but ones that are really bad at catching moths. And so when we're looking at it, you know, this bolus spider, Placylobus. So Placylobus is cool. They come in all these weird colors. They're like bright blues and purples and also browns. There's like a weird mixture of them, but their bodies are always these strange shapes. Then you have Pseudarachne, which is, you know, this is also a spider, even though it looks like a, you know, like a snail. And then that is, you know, the traditional spider that we're used to. And so when we look at here, you know, even though, you know, this bolus, this excavata is not producing silk, is not producing it, we can still show that this is monophyletic for pheromones, right? They're the only species that use pheromones in order to catch moths. And so this would be a monophyletic group. If you then include these placillivus spiders, you know, you're including this species here, but you're not including Pseudorachne or any of these other descendants. That would be a paraphyletic group. And then this polyphyletic group would be, you know, including all of these spiders here, but not including their common ancestor or something of that nature. And so, yeah, and so in this case, we have what's known as a shared ancestral character. And so that's a character trait that originated from an ancestor. And so, for instance, a shared ancestral character of all these organisms would be the creation of silk, right? All of these organisms can make silk in some type of way, and that evolved from some original gene that their ancestors had that either produced silk or some type of silk-like protein that was changed, right? And so here's just a little bit of my research. You know, when spider silk, when spider glue flows, for instance, this is what happens on glass. You have silk that comes into contact, it spreads like water, it goes out, it's just spreading like a glue droplet, right? When the same glue droplet, so this is that species I was telling you about, comes into contact with moths, look what happens. It spreads further, it spreads everywhere. And one thing I haven't shown you on this is actually 1/10 the time as this one. So this is spreading 10 times as fast. Also, if you look at this, the glue flows and then it actually goes underneath the scales and you can see an air bubble that pops up. So instead of sitting on top, it's actually flowing underneath and forcing air out and gluing all these scales together. When you look at it from the side, you can see that it spreads really, really far. It doesn't just spread during that screen, right? It's going to spread this huge distance. It's actually going to go underneath the scales. This part here, it's actually going to fill in. And so this glue is specifically evolved to interact with the scales in a way that lets them flow beneath them. And so that's part of my research, is figuring out what properties you need in order to spread on stuff like this. Because if you think about it, you know, since these scales are kind of falling off very readily, that means that it's kind of like a dirty surface, right? It's like if you're trying to take a poster and you're trying to put it on a wall that has dirt or something on it, the glue or tape that you use is going to get gunked up and then that poster is going to fall right off, right? And so that's kind of what's happening with these scales, right? The glue tries to spread on the scales inhibit the adhesion to the wall, and then the moth just escapes in this case. And so, you know, we have this thing where our glue droplets are spreading on the moth scales. They're spreading over them and under them. I'm going to skip this a little bit just because I was late. Right. And so the idea of these genes evolving is that an organism's evolutionary history is documented in its genome, right? It's documented in those changes that we notice in sequences from ancestral forms. And so what happens is that, you know, you have some type of ancestral gene that exists in a population, in a species. What happens is that as speciation occurs, you have divergence in any particular gene, right? As your birds start to evolve, as they start to choose different mates, you're going to have genetic drift. You're going to have all of these other factors that are going to change genomes. You're going to have, say, for instance, you know, a species of spider where all of a sudden that silk gene is no longer going to be that consistent, you know, tacky glue, right? it's going to evolve to slightly change its properties. And so that glue is going to be a lot more flowy. It's going to be able to spread on those moths. And so these are called orthogonal genes, which are genes that came from the same ancestral gene, but have since diversified and changed. And so when we're comparing, this is what we're doing, right? We're looking at, say, your 16S DNA. We know what it should have been a long time ago because we have ancient bacteria. And we know that it's changed in species A by removing one, moving one nucleotide. And we know it's changed in species B by changing a different nucleotide, right? So we're comparing these changes over time between species. And so we do this through morphological or, comparisons, but we can also do this over huge genomes, right? We can take something like that 16S, which is, again, very highly conserved in most organisms, not just bacteria, and we can backtrack that. And that's why we're able to say compare between animals and fungi, right? We have 4 times as many genes as yeast, but we share, 99% of our genes with mice, and 50% of those genes are overlaps between yeast and us, right? We have a lot more DNA than them, but we still have a lot of those same sequences evolutionarily. So we find an old enough gene to compare to, we can compare between very distant species. Because again, we're very different from yeast, but over millions and millions of years, those changes in fundamental proteins should only happen, one nucleotide here, one nucleotide here, one sequence here. Because, small changes usually don't kill an organism. When you have huge changes, you're going to have, fundamental differences in their proteins and things like that. So not all genes are necessarily that much more complex. It depends on how they change. Different parts of our DNA appear to be more or less changeable compared to other ones. Also, certain species seem to evolve at a higher rate. So, for instance, dogs are considered having very high genomic plasticity. If you think about, how quickly we have selectively bred dogs to be all of these different morphs and physical types compared to, say, for instance, cats. How many different body plans on domesticated cats are there compared to dogs, right? We have Chihuahuas, we have Mastiffs, whereas cats are, some are a little bit bigger, but for the most part, obviously, if you know a lot about cats, please let me know if I'm ignorant on this, but, they're physically not as diverse, right? So certain species have the ability to change a little bit more. They have this plasticity in their DNA. And so that makes this idea of looking at these gene sequences a little bit more complicated, but it's just something we need to know. And so you have these genes that essentially result from duplication. So for instance, this first one, right, these orthogonal genes are genes that have slowly changed. They're essentially homologies, right? This could be a gene for the tibia or the fibia or some type of mammal bone, right? And small changes will change how those evolve. But you could also have a lot of the ways that these, changes occur without killing an organism is these paralogous genes, right? So these genes which have somehow duplicated, right, during the evolution of them, or during cell DNA replication, somehow you actually created a duplicate of that gene. So now you have 2 versions of that same gene existing in your body. And now that you have a duplicate of it, there's a chance that over time, each of those will slowly change, if that makes sense, right? You're going to have a point mutation in copy A, but then, millions of years later, a different organism is all of a sudden going to get another small change in that genome, right? Except it's going to be in a different copy. So now those completely same genes have started to diverge from one another and started to specialize. And so this is how you can have changes in these genomes. So for instance, spider silk, extremely repetitive gene sequence. It is One of the reasons we are having a hard time making spider silk, so for instance, we tried to put it into other protein producing organisms. Like if you ever heard of spider goats, they did a thing where they tried to take spider silk and have goats produce the proteins in their milk. And so what we would do is we take that goat milk, we'd spin out all of the spider silk proteins, and then we would try to use that to make threads, right? Spider silk is extremely tough, super strong. One thing we've noticed when we tried to do that, though, is that the silk is coming out very, very weak, and we don't understand why. Turns out that spiders don't just have one copy of their silk genes. They have 100 replicates of the exact same gene. And so we don't understand if when genes are being expressed, if you need all 100 copies of that, or maybe you only need 50 copies for the silk to come out well. But the point is that spider silk has evolved all of these different versions of that same silk gene. And then in certain cases, it's evolved to change. And so spiders, say the ones that make webs outside, can produce 7 different types of silk. They all have very similar bases, but their repetition of particular amino acid sequences is different. And so all of their mechanical properties are different. So when you see a spider web outside, that's actually produced from 5 different types of silk. You have the radial silk that makes the frame. You have the capture silk that makes the spiral. Then that glue that I study is actually a different type of silk that's also produced by a spider. So that's a 4th type. Then you have a special anchor glue. Then you have silk that the spider uses to make egg sacs and wrap prey, right? So all of these are very similar chemicals that at some point were probably the same gene. And then over time, over different generations and different lineages, that gene got copied, and then slowly but surely those sequences changed. And so now spiders can produce a silk that's primarily made of beta helices and alpha spirals. And so it's super stretchy, and that's how the capture spiraler is made. And then they can also make one that's almost entirely glycine, and so it's extremely stiff. And so that's what it uses to make egg sacs and to wrap up prey, right? So in these cases, you had these same genes that have slowly devolved and changed. And so this doesn't only happen in spiders, right? We can take any ancestral gene, such as, again, that 16S, and depending on the species, there's possibilities that 16S was replicated and changed over time. And so, spider silks have these different properties. And so genes are repeated, leading to different silk types. And these copies then diverge. And so depending on what species you're looking at, you may have to find some ancestral gene that is shared between them, but you should be able to look at this divergence. And again, this happens more or less in certain species. Yeah. All right, so I'm going to skip this just because we're running a little bit low on time since I was late. But if you'd like to learn about the strongest spider silk that there is on Earth, this is the Darwin bark spider. So this is a spider that's about the size of a quarter, and it makes webs that are somewhere around 25 to 70 feet long. In Madagascar, these spiders make silk webs that are about 3 feet high and can be anywhere between 20 and 70 feet long over rivers, right? So scientists were like, this is the craziest type of silk I've ever seen, use of silk. So we're gonna run into Madagascar where they're found and see and test their silk. And it turns out that they have the toughest silk, right? They've evolved in this very particular environment to make really long strands. And so this is kind of the idea of, scientists call this like bioprospecting. They look at really species that are in really intense evolutionary pressure in an environment, and then they look at them to see what kind of things might have evolved under that pressure. So for instance, all spiders are using silk. A lot of them are making webs. This is the one that's making the biggest web. It probably has different properties. But what scientists do is they try to do this idea of what's called molecular clocks, right? So what they're trying to do is they're trying to track how long it's been since species diverged and how long these different changes in genomes take to occur. And so depending on the location of the gene, depending on what kind of gene and species, there are different estimates for this. But generally speaking, something around 25 million years, for instance, in this particular gene, it takes for one of these sequences to occur. So we have this ancestral gene here. After 25 million years, species A has changed, say, this one part to a G, and then species B has changed this last G to a C. And so we have this ancestral form, again, 16S. And so we can kind of estimate based on how many pair changes there are, how long they diverged from one another. And so, for instance, here you have a gene that's the same, held by two species. You have these two separate mutations. And so one mutation will create one gene that just continues to change over time, leading to species A. But then on this one, species B duplicated this gene by accident. And then each of those genes started to diverge. And so you have a change, and this is how you get species B and C from a common ancestor that was shared with A. And so this is an approach for measuring the absolute amount of time of evolutionary history. Again, as I said, different things make this more or less difficult. For instance, those fish that were really closely related became one species after only 4,000 years. Beetles, for instance, haven't really changed much in 40 million years, right? And so the more similar two sequences are and the genomes are of two species, the less time we assume has been passed down, right? Because we're assuming that positive and non-detrimental mutations are occurring periodically and over a set time. Obviously, something can happen. You can have, I can't remember if Kelly or Dr. Strauss talked about this, Professor Duncan or Dr. Strauss, but there could be these huge deletions. There could be different things in the environment that can lead to genetic change, like mutagens. But for the most part, we're going to assume some natural order of natural selection. And so different genes can evolve at different rates, even in the same evolutionary lineage, right? So if you have a really, really long gene, you are more likely to have a mutation occur in there, right? There's just more space for it to mess up. Also, depending on if a gene is very, very dependent on introns versus extrons, right? If you have a gene that's changing in an intron, it's not really going to matter very much, right? And so genes will evolve at different rates, even in the same organism. So, the gene that is coding for the capture glue might be very different in its evolutionary process, say the silk that's being evolved in order to wrap up prey or things like that. And so the number of nucleotide substitutions in the orthogonalist gene is proportional to the time that has passed since the genes branched, right? Or at least that is the assumption. We are hoping, and because a lot of this is based on fossils, is based on ancestral species, 25 million years to change versus 23 million years to change, depending on the context, might not matter that much. But again, trying to figure out how closely related species are, we can do it at least in some way through this. And so when we're talking about phylogenetic trees and maximum parsimony, right, what we're trying to do is figure out how these species are created in a way that minimizes these genetic changes, right? Each organism in this tree is not just gaining a new trait or changing. It is the process of this. It is this long change in nucleotide sequences, insertions, and deletions over multiple species. And so this is just like, again, when we look at a common ancestor, we're looking at an organism that existed back here or over here, and had fewer changes in mutations, and that hopefully we can correlate and kind of look at those mutations over time. And so last thing I'll talk about is, I love my complications of biological concepts, but, for instance, there are certain things, talking about viruses or always going back to bacteria, how asexual species really mess up our understanding of evolution. We have this idea of horizontal gene transfer. And so it is when you use, say, in our case, we can do things such as using viruses for this or things like that, but it's when you have substantial movements of genes between organisms in different domains. So for instance, this is the idea that we can use gene therapy, right? So one thing we do is viruses, if you don't know, have their own genetic information. They're not living, but they can actually inject themselves into your cells, and then they use your machinery in order to create their own, important proteins. And so what they do is they actually inject their DNA and attach it to your DNA. And so your cell starts producing the ribosome, starts using your ribosomes in order to produce the appropriate, proteins and chemicals that you need for this virus to survive. And so scientists have been doing a thing where they actually program a virus with a new gene. And so that virus goes into your cell membrane, goes into this different vascular package, and then actually gets shipped into your nucleus. And then it will insert that new gene into your DNA. And so scientists are trying to do this thing where, say, gene therapy, where you take a healthy cell replication inhibitor and you inject that into someone who has cancer. And so the idea is that the virus will reprogram that cell with the new DNA, and hopefully that will help the cell, stop replicating in a cancerous way, right? Or in this case, a big thing that this happens is with bacteria. Bacteria can share genomes. They can like take in plasmids from other ones. They can, a virus could transfer theoretically a antibiotic resistant gene to another one. And so, there's always this possibility that when we're looking at, say, different types of species, say, for instance, bacteria, and we're looking at their evolution, that genomic clock might not work, right? It might not have been a change of, one nucleotide at a time, right? It could have been an evolution of that nucleotide in a different species. And then that species somehow, say, bacteria dies and it gets absorbed by another bacteria. Boom. There's a brand new gene that is available in that population that didn't exist, that did not evolve from a single sequence in changes in nucleotides, right? And so when we look at these certain things, we need to look at, think about those ideas as well, right? For instance, there's a type of snail, sea slug that actually takes in the chloroplasts of organisms that it eats, and it utilizes that and incorporates it into its own body, right? They're sharing genomes, and there's possibility that we have gene transfer between these different species as they're interacting. And so, for instance, doctors are using viruses in order to change genomes of patients with secure disorders, right? So that's all I have for you. I was going to play a video on the gene transfer if you have time to watch it while we're here. But that is the end of today, and I'm sorry that I was so late.
To say that your therapy has the potential to be curative is huge.
This is a particularly exciting time for gene therapy because the promise and the potential that has existed for 30 years in the field is now starting to finally bear fruit. AAVs will allow us to address a number of different diseases because of their ability to deliver genes to a number of different tissues inside the body.
With real estate, it's about location, location, location. With gene therapy, it's about delivery to a specific location where you need to restore the function of that gene. Adeno-associated viruses, or AAVs, are small microscopic virus particles that can deliver DNA into cells. They have a protective outer shell on them, which consists of viral proteins. Those proteins contain the molecular addresses to target certain cell types and tissues for delivery of your therapeutic construct.
There are some advantages for using AAV over other types of viruses. One is that they are not known to cause any human disease, and that improves the safety profile. The second of which is that they can be directed to infect a variety of different organs in the body, which makes them more broadly amenable to gene therapy applications.
Typically, AAV vectors are used to replace a defective gene that's found in a genetic disorder.
The real promise of gene therapy is to actually correct at the genomic level the cause of disease, which could in fact result in a lifelong cure without the need to take pills every day, to have painful injections every day.
Within Novartis, we're currently developing local applications for AAV vector medicines, primarily in neuroscience as well as in ophthalmology.
Novartis is building its AAV capabilities by using a combination of internal innovation as well as external partnerships. We've built teams internally that are now making AAV vectors. We're also looking externally by finding partners that may have been able to innovate the biology outside of our walls and bringing that internally to be able to really drive the innovation in the space.
We've invested in the infrastructure needed for research, for preclinical development, for clinical development, and most importantly, manufacturing in order to provide these innovative medicines to patients. This is a very exciting time for gene therapy.
Cool. And not only are we learning about evolution, but we're learning how to make our world better and help people. So sweet. All right, that's all I have for you today. I will see you all on Wednesday. Thank you for coming.
Would you just note when the test is going to be, or have you not decided yet?
I will look at it today. I haven't looked at what the results were. Last time I checked, it was pretty 50-50. So I'll send out an email / let you know definitely by Wednesday.
Thank you.
Thank you.
Thank you.